Good morning. This is Dr. Dan Guerra coming to you from VerEvMed Studios. I'm going to give you a talk today about lipid profiling of schizophrenia. So we'll just go ahead and get started. From the beginning. There we are. All right. So that's me over there, Dr. Dan Guerra, and I guess I'm over here in this video as well. My email is down there, and this particular presentation is coming to you from two organizations which I own or co-own. One is VerEvMed, which I've been producing a lot of videos for the last over almost a year and a half, and then Authentic Biochemistry, which is also my podcast. So let's just get started. I guess just as a preliminary, my background is I have a PhD in biochemistry, and my background is basically in lipid metabolism. Worked in plants for a while, and then I'd worked for a very long time in animal systems and ended up working in neuroscience for the last 10 years or so. So I've been a professor at a couple of universities across this country, and now I make my living working with my company, VerEvMed, as a consulting firm. And we can talk about that some other time. This is a free lecture, and I would like to get that out there as soon as possible. So let's go ahead. So, all right. So let's hide this if we can. All right, that's my Facebook page down there. And all of these video lectures you're going to find in that Facebook page, as well as a lot of other information associated with VerEvMed, increasingly about authentic biochemistry. You can see VerEvMed from a website, which is www.verevmed.com, or you can access it through my Facebook page. And the authentic biochemistry you can pick up on any of your favorite platforms, including iTunes and Stitcher. Okay, so let's just get started. Now, I'm going to start off looking at, and this will be the basic paper we're going to take a look at today. Now, what I do in VerEvMed is I go into the primary research and pull a paper out, or pull several papers out usually, and we dissect them, we analyze them. We look at the introduction, we look at the methods and materials, we look at the experimental design, then we look at the data. We decide whether or not the data looks like evidence, and then we try to verify the evidence along with the utilization of the discussion that's usually in the manuscript. All the papers that I ever cover are always in peer-reviewed journals. I don't deal with anything from popular press or anything in popular science. These are all bona fide, peer-reviewed publications. We tend to work with papers that have recently been published, like this one published about a year ago. But sometimes we do go back and look at papers that are more aged. And we go back and look at those because there's nothing wrong with papers that were published several years ago. I mean, there's something wrong with them now. There was something wrong with them when they were published. That's what I'm trying to say. But as you know, research is such that every time we move a little bit forward in science, we tend to learn more and we get a better understanding, therefore a better story of what we think is happening in whatever complex system we're studying. And in my field, I'm a lipid biochemist. So we are more or less regularly increasing the largesse of our knowledge base in lipids. And unfortunately, also, there are a lot of papers published daily, weekly, monthly, etc., that don't really contribute too much to the primary literature for various reasons. And it's usually not because of incompetency. I think it has a lot to do with people not really reading the literature. And then moving forward, doing science, publishing that science, and continuing not to read the literature. And that's what I would like to straighten out. I'd like to get people to read more, particularly medical doctors, people in the health professions, but also colleague scientists, or anybody that wants to say that they know something about a given scientific field. Again, I cover molecular medicine, and primarily I look at lipids, but I also look, of course, at DNA, RNA, protein, carbohydrate metabolism, and utilization in the eukaryotic system. So let's just get started. Sorry for that long-winded introduction, but I realized I kind of had to say it. So this paper published in Metabolomics a year ago.
Let's take a look at what they're saying. First of all, we know that schizophrenia is a complex neuropsychiatric disease. However, it has occult endophenotypic pathologies. We don't really know if there's some kind of link, a neural association that we can pinpoint that's going to have a biochemical, and cellular basis that will say, yes, if these lesions are found in these pathways and in these cell types, or these cells are degrading more rapidly, you're always going to get this given disease. Very, very common in neuropsychiatric conditions where any one gene or any one network of systems doesn't really explain the presentation of the disease, the clinic. This actually happens not just with neuropsychiatric diseases, but diseases in general, cancer, cardiovascular disease. They all sort of fall into this, although in cancer and cardiovascular disease, there's been more time spent using animal models, and therefore we come away thinking that we know more about the disease. But as I'm going to mention later, animal models do not depict human disease. They only are models. And I think we need to move away from animal models and try to work more on the human system, particularly in neuropsychiatry. Okay, so there are candidate associated what I call molecular patterns. That includes expression of genes like proteins, but also receptors that are found in the membrane, how they're found in the membrane, signaling molecules, signal transduction pathways, the whole business, as well as, of course, tandem gene expression, and then epigenetic modifications of DNA, and thus alteration of RNA that causes differential gene expression. All of that. There are candidate-associated molecular patterns, and they may drive or result from the degeneration of the central nervous system, neuronal synaptic structure. So it looks like something in schizophrenia has to do with some kind of degeneration. Now, basically all neurological diseases have something to do with degeneration, unless they're cancers, and then you're talking about a proliferation of cells in the form of a tumor. And even then, eventually, you get a lot of degeneration. Now, metabolomic studies have identified lipid metabolism as a potential target. So we need to focus in on that. That's why I wanted to look at this paper. So lipids as a specific metabolic pathway, as opposed to, say, nucleic acids, proteins, or carbohydrates. So this paper is going to compare treated versus non-antipsychotic treated schizophrenics, human patients. Actually, these are going to be dealing with postmortem studies, versus control subjects. And they're going to be looking at a potential neuropsychiatric serum lipid profile and see whether or not they can link this to people that have schizophrenia that are or are not on antipsychotic drugs, and determining whether or not the antipsychotic drug, of course, has an effect on that serum lipid marker. So what are we talking about when we're talking about schizophrenia? Well, I looked it up in DSM-5, and it's quoted here at the top. Schizophrenia is characterized by delusions, hallucinations, disorganized speech, behavior, and other symptoms that cause social or occupational dysfunction. For a diagnosis, symptoms must be present for 6 months and include at least 1 month of active symptoms. Okay. The DSM-5 requires that schizophrenic needs to present with at least 2 of those symptoms you see up there. As with the majority of neuropsychiatric disorders, there is not a unique biochemical or genetic biomarker for schizophrenia. Rather, there's a somewhat what I call subjective clinical diagnosis, including a standard constellation of symptoms, which we just looked at there. Of course, some kind of dysfunction at the social, familiar, career, phenomenological level. So, according to the Diagnostic and Statistical Manual of Mental Disorders, I need to move this here. The lifetime prevalence of schizophrenia is approximately 0.3 to 0.7%. So it is significant, but it's substantially lower than other kinds of neuropsychiatric diseases, for example, like major depressive disorder. The psychotic features of the disorder typically emerge between mid-teens and mid-30s. So it's a disease that unfortunately afflicts the young. Peak onset is first psychotic episode is usually early to mid-20s for males and slightly more older in females, late 20s.
So it's not a disease that you see come up. Schizophrenia doesn't come up when a person's like in their 50s or 60s, although there can be psychotic breaks, which may be associated with schizophrenia that otherwise was occult or latent. So how they do the work: plasma and tissue swab technologies for protein and other metabolite-based biomarker discovery has been in vogue for a long time, because those kinds of things, like drawing blood or doing a tissue swab, and then from that tissue swab taking cells, from those cells doing extractions, or running the cells through, say, a flow cytometer. Why is that done? Because it's non-invasive, basically, except, of course, drawing blood. Some people don't like that. But it's not like going and doing a biopsy, you see. Yeah, it's also relatively cheap. You can do it right there in the clinic. Relatively enriched in potential meaningful variations in quality and quantity of biochemical structures. Cerebrospinal fluid has also been tapped as a source of these metabolomic profiles, as well as muscle punch biopsies, which are a little bit more invasive, of course, and were available post-surgical extraction of ablated tumors, cell masses, and damaged tissue organs. Of course, as I said, we also have post-mortem tissues. These metabolic experiments are becoming more fashionable, and medical interventions, especially in neuropsychiatry, with areas they adapted to positive negative control studies by using disease versus healthy subjects, and during the administration and course of therapeutics, such as, of course, using pharmacological agents, such as drugs that are specifically associated with that neuropsychiatric disease. You also have psychological and psychiatric analyses. Lifestyle changes, such as exercise and diet modification, are all thrown into the big pot in therapy for neuropsychiatric disease. Now, in the case of atypical antipsychotic agents like olanzapine, or risperidone, or aripiprazole, and Concerto for diagnosed schizophrenics, a lipidomic profile has been variously published with, if not robustly accurate, at least naively purposively approaches to define a baseline of common lipid panels. So you see what I'm saying there. People have tried to use antipsychotic agents. Some of these are the atypical antipsychotic agents. Atypical because the typical ones were already tried, they didn't work. And then going ahead and looking at things like lipid profiles. And so they see a lipid profile, they think, oh, wow, look, it's different from the baseline. Maybe there's something to do with the disease. What I'm saying is that they're naively purposive approaches. That means that they think that they have an actual purposive idea that this will function as a biomarker, as an associated biomarker, but it's not always the case that they've proven that. Okay, so the metabolic or metabolomic analyses can be fulfilled through untargeted or targeted approaches. You can use one of two ways. Two strategies are often deployed by liquid chromatography, mass spec, and usually it's based on untargeted metabolomics. It's usually the one because that's considered unbiased, you see. So you have two different kinds: data-dependent acquisition or data-independent acquisition. What follows I'm going to show you is a summary of schizophrenia, DIA-LC mass spec. Okay, so it's going to be data-independent acquisition. It's going to run the LC mass spec to generate lipidomics, and where comparisons are drawn among naive schizophrenic patients, antipsychotic treated patients, and healthy subjects, and for purposes of a biomarker candidate identification and potentially a pharmacotherapeutic modification of lipid metabolisms observed from serum lipid profiles, biomarker screening, and of course pathological studies, as well as antipsychotic development and mechanistic studies, which goes back into usually animal studies, right? Rodents. All right, so now this gives you kind of a brief overview of what we mean by doing mass spec, LC mass spec. This shows you the liquid chromatography, and it shows you the mass spec. There's two different types. We're trying to show you that the one that they are using might be a little bit better than what is commonly done. Okay, so this is a schematic for data-dependent and data-independent workflows using a mass spec time of flight machine. So what you've got here is you've got these sample analytes, okay, during a high-resolution data-dependent acquisition.
A mass spectral scan, usually on the order between 4 and 1200, that's mass per charge divided by charge ratio. So within that range, so that would be like a molecular ion of the precursor ion, the molecular ion, is performed in the first quadrupole, like shown here. These are all the metabolites coming off the LC, right? They're all run through this particular isolation in this window between 4 and 1200 mass per charge ratio fragments, right? And then ion abundances above a set threshold are selectively collided with nitrogen in the Q2 phase. This is the second mass spec here, okay? That generates a second scan, all right? So there's your second scan there, right? So you have this first scan, which you have a lot of fragments with varying amounts and following that mass to charge ratio across that gradient. And then you've got a second mass spec, which is bombarded with another beam. This is with nitrogen. And now you've got the second scans. You've got a more cleared up peptide fragment. This is just showing you for peptides, but of course you can do this with lipids. And then you get detected peak packing. So you've got the molecular ion minus 1, molecular ion minus 2. And so what you're doing here is basically using collision-induced dissociation. And this is collision between the ion and some neutral species. And that results in the conversion of part of the translational energy into an internal energy in that ion and subsequent fragmentation. That's the way mass spec works in general, at least this particular type. Okay. So what else do we want to say here? MS1 filtering and what are called skyline extracts. It's just part of the software here. Peptide precursor ion intensity chromatograms from the MS1. That's the mass spec 1, the Q1 phase. For a given precursor isotopic envelope, and you can follow through down there, following M-1, M-2 from the molecular ion. There's going to be retention time of the corresponding peptide identified from underlying acquired mass spec, and that directs a proper selection procedure and integration of the precursor ion peak, and that's used then for relative quantification. That's what's shown here. It's the relative retention time here, the intensity, and you're looking at the change in the amount of molecular ion after this fragmentation pattern. Now, that's not what's used in this experiment. Here we're doing a data-independent acquisition. Again, it's quantitative, and it can be extracted for a given peptide or lipid precursor ion from the MS1 scan. So you have this MS1 scan here. But now you're running two different mass to charge ratios, okay? 4 to 425 and 975 to 1,000. Okay, so you've got two windows of spectral distribution of these peptide fragments according to that ratio of mass to charge. And then you go into the Q2. So what happens is you get this filtering, and then simultaneously from the second mass spec, okay, the second mass spec then generates what are these blue lines here, okay, which are ultimately going to allow you to separate the initial fragment curve from all the new fragments that are generated. So up here we're talking about these fragments. Now we're able to separate out those individual fragments into this scan densitometry tracing here. So what you get is a stepwise, what they're called swath acquisitions. Again, it's part of the software. Selecting user define is what we want to look at. So the user defines what the precursor ion mass range is going to be. So we can set the window at 425, for example, shown here. Or you can go up 425 to 450 and keep on moving up the ladder. And then all of those fragments go through a time of flight until they get to the detector. Okay, that's the way they work here. That's where this is measured from. And then you get a complex mass spec spectra. Okay. There's the MS1 and MS2 ions. There's just the extract MS2 ions now, showing you those original ions that were generated. And that gives you far more detailed information, and it's independent. That is, you're not isolating just a specific set of fragments, right? So it's data independent acquisition. And what this basically allows, okay, using the CID pathway, is simultaneous and independent measurements of relative metabolite abundance in a single experiment. It just gives you more data, and you're able to get, because especially in something like a lipidomic profile, you're going to need to do this rather than go on a very discrete set of fragments, because you're not going to get as much information, and you have to run the whole experiment over again.
All right, so a little bit about the experimental design. So that's the technique they used. A little bit about the experimental design here. They had patients with no treatment, and then those are called sample group naive. And so you get a result from that group of naive schizophrenics. Then you give them a drug, and you have the sample group treated, and you're going to compare those on a paired t-test between the results of the schizophrenics treated with the drug versus those that are not. Again, it's a DIA-based lipidomic analysis we're doing. And then you have the healthy group. So there's 20 of these schizophrenics, 29 healthy. And there you're just getting a sample group healthy. You're not treating with any drugs, no sham drugs, no placebos, nothing like that. And then you're doing a paired t-test between the results of just the healthy as compared to the combined naive and treated. Okay, so that's the way they did this experiment. That's called the grouped t-test. So what were the results? 9 lipid classes were significantly different between the naive schizophrenic patients and healthy controls. Okay. For example, triacylglycerol, which is a neutral lipid, cholesterol ester, which is a prenyl lipid, and sphingomyelin, which is a sphingolipid, those are 3 different classes, were upregulated in naive schizophrenics. Then you have plasmalogens, which have ether linkages in the one position of the glycerol backbone to the alcohol oxidation state of the fatty acid. So it's a fatty alcohol. So you have plasminoyl phosphotylcholine, plasminoyl phosphatidylethanolamine, and they also have the cholesterol ester, phosphatidylcholine, lysophosphatidylcholine, and lysophosphatidylethanolamine. These are going to be just glycerol lipids, ester linkages, not ether linkage. And also acylcarnitine. All of those, okay, look like they were downregulated. Now, okay, so we have to discuss a little bit about where all these lipids fall into pathways. That's what I'm going to show you in a moment. Additional lipid classes were significantly downregulated after the antipsychotic drugs were administered. And those included cholesterol ester, triacylglycerol, free fatty acid, glycerol cerebrosides, phosphotylcholine, this plasmalogen with the choline in the 3 position. And then sphingomyelin shows up as the ceramide, which is another sphingolipid. So that's interesting. So it looks like the drug had some effect on all these lipids. Sounds like maybe we're getting somewhere, right? Maybe. Finally, triacylglycerol and cholesterol esters were upregulated in naive schizophrenic patients and downregulated after treatment. So, okay, they were upregulated when you don't treat, and then when you do treat, those 2 signals drop down. So that's also rather interesting, or could be. Whether or not it has any sense, we look at the pathways or we look at the whole background of how these experiments are done, you'll soon see may differ from saying that there is significance. Okay, so downregulated naive schizophrenic patients where antipsychotics strengthened the decrease of plasminoyl phosphotylcholine, PC, and lyso-PC. So let's take a look at these pathways. Just standard plasmalogen biosynthesis. Start off with the fatty acyl-CoA. This is now dihydroxyacetone phosphate, right? So that's coming directly off of glycolysis. And so you combine those, you make a 1-acyl dihydroxyacetone 3-phosphate. Now you're going to go ahead and take a fatty acid, right? And look what you've done. Now you've taken any other fatty acid. You've generated a fatty alcohol, okay? So you've converted this fatty acid now to an alcohol, right? You change the oxidation state. And you remove the fatty acid from the one position, the ester, and now you've loaded in the alcohol because it's an alcohol. Now it's an alkyl, okay? That hydroxyacetone 3-phosphate synthase. That's the name of the enzyme. And you've got a long chain alcohol now in the one position of the glycerol backbone. You've still got the keto function in the 2 position. And you've got the just a phosphate on the 3rd carbon, right? So now you're going to go ahead and you're going to do a reduction, right? And what you end up with now is you've got an alcohol group in the 2 positions that's going to be ready to get esterified to a fatty acid. See, you got rid of the keto function. Now you got an alcohol function. It's like glycerol now, not like dihydroxyacetone. You've got the ether linkage of the 1 position already made. You just have the phosphate ester down here. So it's a phosphatidic acid. So now what you've got is you add another fatty acid. That's what this is here. Of course, there are always coenzyme A thioesters. Now you've got one alkyl, two acyl. The alkyl means it's a ether linkage there, see? Glycerol 3-phosphate. So that's what's up here. Now you're going to add a head group. So one way of doing that is just pirating the head group after you've already synthesized the phosphatidic acid. And so you're taking ethanolamine, and now you've got one alkyl, two acylglycerol, three phosphoethanolamine, right? And then ultimately you do one more reduction, and you've got the plasmalogen completely designed and set up, okay? So this is what you've got now. So this last reduction gives you a double bond here. So this is going to be a vinyl ether lipid. Okay? All right. So keep in mind that all of these lipids you're talking about that are being measured in this paper are coming from basal metabolism. So what do we mean by that? Well, this slide shows you. Via glycolysis, you make dihydroxyacetone phosphate, and you can make glycerol 3-phosphate, which of course is the backbone for phosphatidic acid and all the phospholipids, right? Also, you can make the neutral triacylglycerol from phosphatidic acid, right? And so from adipose tissue or from the diet, you break down triacylglycerol. You can pick up glycerol that way as well, phosphorylated. Now you're on your way to making phospholipids. You can also generate some free fatty acids, which can be utilized in the liver as acyl-CoAs to build up triacylglycerol from phosphatidic acid, putting in that third fatty acid. Okay, so there's a lot of metabolism that goes on just for the backbone, and this is just showing you the glycerol lipids, not looking at the plasmalogens yet. So there's phosphatidic acid. Removing the phosphate gives you diacylglycerol. Adding the fatty acid makes triacylglycerol. It's just what's called the Kennedy pathway. Building neutral lipid from this phospholipid by removing the phosphate. So phosphatic acid is a very important intermediate for phospholipid synthesis, as well as neutral lipids such as storage triacylglycerol. Now, another way of doing this is taking the entire phosphatidic acid. This is called the nucleotide pathway. So there's multiple pathways to get to these compounds of interest. Okay, so that is very critical to understanding the significance of this paper. You're looking at serum lipids. You're looking at you're being told that because these lipids are in circulation, that it means necessarily that this is a direct conduit to our understanding of the presentation of a neuropsychiatric disease. Now, they're not making any claims about any of these enzymatic pathways or where any of this carbon is coming from. So there's no way of knowing where in this vast array of metabolic sequences you might have a lesion, or if indeed you do, because they're just pulling blood, looking at serum lipids, and not even showing you what fraction are in the serum. In other words, they didn't do a lipoprotein profile, which is what they should have done, in my estimation. Anyway, another way of making phospholipids, you take phosphatidic acid preformed. So this is both ester-linked fatty acids, 1 position, 2 position, glycerol backbone, just the phosphate as on the 3 position. But now you're loading in nucleotide. So you made CDP diacylglycerol, say, and you've driven that reaction by PPI and then 2PI hydrolysis to that. Now you got CDP diacylglycerol against the nucleotide pathway. You react it directly with a sugar like inositol. Now you've got phosphatidylinositol, which is a very important phospholipid in the membrane of eukaryotes, a very important signaling molecule. It generates the phosphatidylinositol cascade, which is linked to calcium flux, kinase cascades, et cetera. Very important. And then you just regenerate CMP, and you build that back up, and you can start over. All right. Now, phosphatidylethanolamin can also be converted directly to PC, phosphatidylcholine, by using acetyl-methionine as the methyl donor. Because indeed, choline is trimethylethanolamine. That's what choline is. So here's the pathway just going directly from ethanolamine, which you can get from serine, okay, after decarboxylation. Ethanolamine can become phosphorylated. That activates it. Then you can turn it into a nucleotide, CDP ethanolamine. See, rather than making the nucleotide with phosphatidic acid, now you've made the nucleotide with the ethanolamine, and now you can react that with diacylglycerol after phosphatidic acid. Phosphohydrolase has worked on phosphatidic acid. You get rid of the CMP. You've activated that ethanolamine. You get rid of this. You get rid of that CMP that activated the head group.
and you add diacylglycerol, and now you've got another way of getting phosphatidylethanolamines. You see there's multiple pathways involved there. Now, those were all glycerol lipids. Here's sphingolipid. Here's sphingosine. Okay? Now, notice that sphingosine has essentially a fatty acid. It has a serine residue. That's where the amino group comes from. That's serine, right? Notice that there's a trans double bond in sphingosine. So how did we get there? That's a trans double bond. Take a look. The way you make sphingolipids, okay, remember some of the lipids they're claiming are significant in a schizophrenic profile are indeed sphingolipids. That's what they're saying, right? So check this out. Here's a de novo pathway for this system. You start off with palmitoyl-CoA, which can come directly from de novo fatty acid synthesis, hydrolysis of triacylglycerol, or simply phospholipase removal of palmitic acid somewhere on a glycerol backbone, and then esterification of coenzyme A, which is also very common. So palmitoyl-CoA reacts directly with serine, and look what happens now. You've driven this reaction by losing CO2, and you've gotten rid of the CoA ester. Now you've made 3-keto-sphingosine. We're building a backbone here. This backbone is going to be the backbone for all the sphingolipids. Now we're going to carry out a reduction. Okay, so now there's keto function more. We've got this alcohol function here. Still got this free amino group. It's going to become important in a minute. Now we're going to take a fatty acyl-CoA, right, and we're going to make an amide linkage to it. So this is amide-linked fatty acids. It's similar, you see, to something like, let's say, diacylglycerol at this point. The difference is, of course, you have an amide linkage there for one of the fatty acids and not a oxygen ester to a carbon moiety. So then you go ahead and look what you've done here. Now you're going to oxidize this lipid, right? And that bond now is going to be turned from a saturated bond, which was originally palmitate, to a trans monoenoic system, trans single double bond monoene. And that's ceramide. That's one of the lipids they say may be important in this paradigm they're trying to describe for profiling schizophrenics. Ceramide can also be acted upon. There are two pathways. One is to react with phosphatidylcholine, removing the entire diacylglycerol moiety and now making sphingomyelin. Okay? So all you've really picked up is the phosphatidylcholine residue, and it's right there. So this is a lot like phosphatidylcholine, only it's not because it's a sphingolipid with a sphingosine base rather than the glycerol base. Got it? All right. All right, classes are not needed. Sphingomyelin, really important because that's the lipid that is going to be acquired from the oligodendrocytes. It's going to myelinate axons associated with those glial cells, right, on those neurons. So remember that schizophrenia, like a lot of neuropsychiatric diseases, have some association with demyelination. Like, okay, a lot of diseases are like this, right? But even the neuropsychiatric, totally occult presentating diseases tend to have some dysregulation of myelination. So this whole pathway, because it leads to sphingomyelin, this is a pathway that's going to be in the oligodendrocytes that are associated with the neurons in the central nervous system. They're going to generate the myelin sheath. When you find levels of ceramide or any of these other sphingolipids in the blood, it could mean any number of things that are happening in these pathways. It doesn't mean, though, that these pathways are happening in situ in the central nervous system. It could be happening anywhere because these lipids are formed everywhere, not just in the central nervous system. Anyway, sphingomyelin, with the utilization of activated sugars, can make gangliosides, and some of them are called cerebrosides, where you can have various sugars attached to this oxygen atom here. And you get rid of the phosphatidylcholine. And these, you can figure out, these were named because these are brain lipids. Glucocerebrosides, for example, sulfatide cerebrosides are going to be sphingolipids you find almost exclusively in the central nervous system. And they play very important functional roles, not just for basically maintaining the integrity of axonal transport of action potentials, right, which is what myelin normally is considered, the whole saltation process that you probably remember from studying basic neurochemistry. But also, all of these sphingolipids act as signaling molecules, and they bind on receptors, changing the differentiation state, the signaling, and sometimes whether or not there's an immune response from any of the cell types that are functioning in the central nervous system, such as the microglia, but also the dendrocytes and also the astrocytes can all signal as immune cells locating, then circulating innate immune cells to come and associate and search for foreign antigens. This is one of the ways that neurodegeneration occurs because of the degradation of the sphingomyelin or any of the cerebrosides, making a more simplified sphingolipid like ceramide, which can induce a pro-inflammatory response, leading to further demyelination, degeneration of neurons. You see, very complicated pathways. All right, this is just the ethanolamine plasmalogen. That's showing you the fact that you've got this vinyl ether, right? You've got this double bond here, so it's been oxidized. There's your ether linkage. There's a fatty acid and an ester linkage, and there is phosphocholine there. Okay, so that is ethanol. That's just ethanolamine. It's not phosphocholine. You need 3 methyl groups there for it to be choline. And then it would be choline plasmalogen and not ethanolamine plasmalogen. Now, there are other lipids that go through, as you might guess, retailoring metabolic pathways so that they can be reutilized directly from the serum to reform sphingolipids or phosphoglycerolipids. And one of them, very famous, essentially ether-linked lipid, is known as platelet activating factor. And that's what it does. It activates platelets and causes clotting in the blood. It's one of the factors involved. So here you can see it's an ether linkage, but it's saturated. So it's not an alkene, like the one we just saw there. It is an alkane, right? Usually a very long chain ether linked alkane there in the one position. Then instead of having a fatty acid, the two position says acetate. That's why it's called PAF acid there. So PAF, platelet activating, and then the A is also for acetate in the two position, acid ether. So PAF acid ether. It's another name for platelet activating factor. I knew you wanted to know that. All right, it's just running you through some of the reactions we already looked at. Phospholipase activity can also work on preformed phospholipids. This one happens to be, you can see the linkage there. This is going to be a most likely a phosphatidic acid based or glycerol based lipid. You phospholipase A2, you get rid of the fatty acid in the 2 position. That fatty acid can go on and do things like become an icosanoid, by the way. Lysyl platelet activating factor acetyltransferase comes in and has an acetate to there. So you had this alcohol group here, right? So that's already an ether linkage. And now you've added acetate. This is one way to make it. So you see directly from a plasmalogen, right? This one has choline. You can, because that's a fatty acid there. So that would be a plasmalogen, choline plasmalogen. You can make platelet activating factor. And so, as I said, all of these potential reactions can occur in cells, but also in the blood itself. Here again, de novo pathways. You're just looking at choline, making phosphatidylcholine, picking up the nucleotide, going through the nucleotide pathway. There's cytidine diphosphocholline, interacts with diacylglycerol, which could be coming from degradation of triacylglycerol after lipase activity, neutral lipase activity. And now you got phosphatidylcholine being generated. And then here's this whole pathway we just looked at, making all of the ether lipids, right? Diadoxyacetone. And by the way, this happens in different subcellular compartments. This is happening in the peroxisome. The rest of these reactions are happening in places like the endoplasmic reticulum, or even some of them in the peroxisome as well. But so what I'm saying is because of the subcellular distribution of these pathways, you have to know where all of these lipids are coming from if you're going to suggest that when they're found eventually in the serum that they're affecting metabolism in, let's say, the oligodendrocyte. And so you go all the way back to the cell where these lipids are made, and there's no signature that says where they're made or how they're metabolized or how many times they've turned over. So these kinds of broad-based profiling using mass spec that just look at serum lipids doesn't really tell you anything about the origin of the lipid you're looking at. If you don't know the origin, how do you know which component of this pathway is being affected? Right? Even a good lipid biochemist is going to need a lot more evidence to tell you that, even though we know these pathways. It's just showing you, again, the de novo synthesis of all the sphingolipid pathways. All right. Here's ceramide going to sphingomyelin cerebroside again. Here's a complex glucocerebroside. You see there's a lot of different sugar moieties that can be added. So this is GalNAc, N-acetylglucosamine, galactosamine, very important sphingolipid class of the glucocerebrosidase pathway in the central nervous system in humans. Okay, so I just want to remind you during digestion, when you take in lipids, that a lot of the lipid degradation is occurring in the intestine, and particularly with lipids, that's important. And you build up this first lipoprotein called the chylomicron, which its origin is indeed in the small intestine. And the chylomicron requires various apolipoproteins like this B48 in order to combine with all the lipids that are coming in from digestion. Long-chain fatty acids making acyl-CoAs, esterifying cholesterol, making cholesterol ester, cholesterol from the diet, triacylglycerol being broken down or being resynthesized after being broken down by getting rid of the fatty acids that you came with. The reason that lipase-mediated breakdown of dietary triacylglycerol occurs is because often the component triacylglycerols that you take in the diet are variable in terms of chain length. And what the small intestine tries to do is make those non-variable so that triacylglycerol can be ready for being either a biofuel or for manufacturing complex lipids like phospholipids and sphingolipids in the liver later. All right, so this is the first real apolipoprotein made, the chylomicron. Now notice you need amino acids to make the apolipoproteins. So protein degradation from the diet is going to generate the right amino acid pools that is going to carry out what? You're going to get translation of the apolipoprotein B48 in the small intestine, right? And when that happens, it's going to need the amino acids, and the transfer RNAs are going to be isoaccepting those amino acids and being charged with them, so you make the right apolipoprotein fraction. And if you don't have the right protein in the diet, you won't have the right composition of amino acids. So that can cause a problem with chylomicron production, which can cause dyslipidemia. Just saying. This is just showing you how HDL traffics. You hear a lot of things about lipoproteins, and you're told that HDL is the, I hate this word, good lipoprotein, and LDL is the bad one. I mean, those words have nothing to do with, there's no valency about any of the lipoproteins being, quote, good or bad or indifferent. There's no real modality to them at all. All they are different fractions of lipid and protein agglomerations, non-covalent associations of lipid and protein. They go into circulation because lipids are not soluble in the aqueous. So you can see here that HDL can be synthesized directly. I told you various fractions of these lipids are interchangeable, made in the hepatocyte. HDL can leave the same way that a very low density lipoprotein can leave. It's going to be picking up different apolipoproteins and different phospholipids. But once HDL is out there, it can interact with other lipoproteins. And when it does so, it's going to be transferring and mobilizing other fatty acids associated with the lipoprotein fraction. And all those things are going to be picked up various places in the periphery. But periphery is very good with LDL receptors because it needs to get cholesterol from the liver, because it's either going to come from the diet or from cholesterologenesis, which occurs in the liver. Cholesterol is important for all the biomembranes in the body. So you've got all this interaction with HDL interacting with very low density lipoprotein, generating, again, more lipoproteins. There's the intermediate density lipoprotein, picks up another apolipoprotein fraction, or picks up or loses glycerol, and now you're back to LDL. And now the LDL can go into macrophages, and that can actually lead to atherosclerotic plaque if that's an oxy-LDL. That's where some of the indication that you get from the pathology of this disease. Of course, you also get direct uptake of fatty acids from the CD36 pathway, which I talked about before, and muscle cells, and fatty acids can be used in beta oxidation to generate energy for muscle contraction. Chylomicron remnants can make it back into the liver, and they can reassociate, pick up other apolipoproteins, and they can reform chylomicron remnants, or they can make LDL. They can be packaged in the endosomes, and they can go through this pathway where they leave again the liver. This is called retro trafficking of lipoproteins back into the liver. So the liver is really an important station for lipoprotein remobilization, resynthesis back to the periphery. Periphery includes the adipocyte, muscle cell, the macrophage, and anything in periphery. In fact, even in the brain, there are lipoproteins that move up into the brain. They don't really cross the blood-brain barrier, but partial degradation of them does allow for things like ApoE4
to cross the blood-brain barrier, and that's actually associated with Alzheimer's disease. So what do you think of that? And that comes from HDL. And there's where the intestine is doing the manufacture of the chylomicrons that we saw blown up in the last slide. All right, I just wanted to give you that overview of lipids. So let's move on real quickly here. Using the molecular pathways as a guide, right, what we just looked at. What can be suggested from the research we just looked at? Because cholesterol esters and triacylglycerol were upregulated in naive patients, and they were downregulated by antipsychotics in schizophrenic patients, there's no correlation to the disease stage, okay? Both of those lipids are trafficked on several lipoprotein fractions, as you just saw. So since these were not associated with HDL, that is, they didn't do experiments to associate them, they could have, LDL, oxy-LDL, VLDL, IDL, or any other key classes, any trends remain occult as to dynamic molecular events or disease ontology. So you're just looking at end results. So you have no way of knowing if, because you're accumulating given lipid fractions, if they are even mildly associated with something as complex as schizophrenia. Secondly, whereas plasmalogen, phosphatidylcholine, PC itself, and lyso-PC were downregulated in the naive schizophrenics and further downregulated by drug treatment, which is sort of interesting, right? Because you would think that drug treatment would flip that lipid profile. Didn't it? Just decreased it more from the healthy people. See, as compared to the non-treated schizophrenics, without enzymatic activities and vectorial direction of de novo and retailoring pathways, the ones I just showed you, among these lipid species, no coherent hypothesis can be deduced from the data. I don't see any hypothesis you can get from here. You can't make any kind of hypothetical deduction for any way of deciding if these results can be put into the paradigm consensus largesse of understanding of this pathway in terms of endophenotypic biochemistry. So, while some lipids were preferentially downregulated after drug treatment, the cerebrosides, ceramide, and free fatty acid, compared with non-treated schizophrenics, these three lipid fractions shown there have no coherent network without some analysis of flux through the pathways and cell types from which the serum lipids were generated. This, coupled with no lipoprotein or receptor data in this paper, render it study ineffective and inconclusive, I'm afraid. There is no event ontology here. All you're looking at is substances in the serum, right? Therefore, no hypothesis can be deduced at all. That's my problem with this paper. Nothing wrong with the work, but it doesn't really tell us anything about lipid profiles in schizophrenics, in my view. So the dialectic, very simple as this. This is a dialectic thesis. Serum-based lipid profiles may reveal associations between cellular metabolism and schizophrenia. Okay. Antithesis. Serum-based lipids may not reveal associations. Okay. Synthesis. Remember, you're carrying out a dialectic. This is an opinion. This is going through the hard mechanism of trying to find truth using logical alteration of premises and within theses and antitheses so that we can discover what is true in statements we're calling premises to generate conclusions, ultimately, in a sound logical argument, for example, which all science should be based on, right? So the synthesis here, after looking at these two, is results indicate that the experimental design was not sufficient to link differences with either disease state, severity, pharmacotherapy, or resolution. Sorry. Now, one more paper real quick. This is from the Asian Journal of Psychiatry. Not a paper I read a lot, but there's nothing wrong with it. And look at what this paper was just published, right? Late April of 2019. In fact, this is an EPUB ahead, so it probably won't come out until next month, June. What did they talk about in this paper? They say a logistic regression model did not reveal any significant association between adiponectin, which is a specific kind of adipokine, levels and the metabolic syndrome in schizophrenia patients. Because previously, people have suggested that adipokine levels are associated with metabolic syndrome, which is comorbid with some schizophrenics. You see, this is where they were coming from. This is after numerous publications linking lowered adiponectin to metabolic disease and schizophrenia. They didn't find it in their paper. While one paper doesn't overturn this one paper, whatever previous work has been done on the subject, to the extent that this current research is conducted with formidable controls, because I've studied the paper, and attempted to rule out bias-associated confounders by appropriate means, it reveals that paradigms and biochemical correlations to neuropsychiatric pathology and or presentation are not easily established or maintained. Okay, so with the last paper, we already, I think, roundly and soundly refuted that we could really come to conclusions. Here's a paper which says all the work that's been suggesting adiponectin levels and metabolic syndrome associated with schizophrenia, this paper did not support that at all. It was well done. So here's the last paper finally we're going to look at. Expression of long, non-coding RNA NEAT1, don't worry about that, I'll explain in a moment, is reduced in schizophrenia and modulates oligodendrocytes transcription. Now see, now here I like this paper more because now we're talking cell types, not just looking at lipids in the serum, right? We're not just looking at adipokines in the serum. We're trying to put something together that's more coherent, three-dimensional with the fourth dimension overriding and continuing everything, which is, of course, time. All right. This paper was published in the MPG Schizophrenia 2019. It was published just a few months ago. All right. So what does it say? First of all, recent papers suggest that oligodendrocyte dysfunction and myelin generation are linked to the pathophysiology of Alzheimer's disease and schizophrenia. Why do I know it's the case? Because I just covered it on VerEvMed. You go back and look at my 1 hour long lecture on that. Okay? Previous VerEvMed video lectures have discussed the role on neutral sphingomyelinase. That's an enzyme that's going to generate ceramide by removing the phosphatidylcholine. That enzyme and ceramide, which is going to be the product of that from sphingomyelin being the substrate, coupled with the switching from glucose to in situ CNS fatty acid beta oxidation, so only the beta oxidation that's in the brain, in accompanying neurons as the basis for the neuropathology of Alzheimer's disease. There was a previous lecture I gave on that, so keep that in mind. This report says that a corruption in oligodendrocyte gene expression leading to cell cycle abnormalities may be the molecular mechanism associated with schizophrenia-linked pathologies. That's what this paper is trying to say. So this is going along this basic research line. So again, it follows a little bit because again, it's looking at cell types in the blood and cell types also in the central nervous system, looking at much more detail than what you get from a standard paper just looking at lipid profiles. So they talk about non-coding RNAs. Those are LNC, long non-coding RNAs greater than 200 nucleotides. So they're not your typical interfering RNA length, which is less than 200 nucleotides. Is why they're called long. They turn over rapidly, and where they turn over rapidly, as an observation, are in glia and in neurons. For example, CNS. NEAT1, which is nuclear enriched abundant transcript 1, is a member of these abundant RNAs. That should be CNS, not CHS. Sorry. The loss of the NEAT expression associates with something called paraspeckle. Don't worry, I didn't make up the term, and I don't like it either, but there it is. It's in the neurophysiology dictionary, lexicon. Paraspeckle is subnuclear structure demolition. So the paraspeckle is basically a subnuclear structure. So when you lose NEAT1 expression, the paraspeckle structure, which is a subnuclear structure inside the nucleus, right, of, for example, a oligodendrocyte, gets destroyed, breaks down. Since paraspeckles act as subnuclear scaffolds for oligodendrocyte necessary proteins, they depend upon NEAT1 for competency, and loss of its expression may then be a part of the endo-biochemistry of schizophrenia. That's what they're getting in this paper. So just to give you an idea how these things function, this comes from a JCB paper from 2009. It says the role of nuclear retention in gene regulation differentiated cells. Look here, there's a gene called the MCAT2 gene. It uses two different promoters. Okay, so there's two different promoters here, right? The transcription. So you're going to get two different splice variants or two different transcripts. You have the MCAT2 messenger RNA, which is exported for translation to the MCAT cation transport protein. Boom, it's exported immediately, and there it is outside of the nucleus. It's ready to be utilized. And then you have this CTN RNA, this longer one, right? And so it's a longer transcript, including the MCAT2. It still has the MCAT2 open reading frame for the protein. the coding region, and it has an extended untranslated repeat at the end, okay? And that contains many inverted repeats. So whenever you see those 3' untranslated ends, that often is something you should take a look at, because that's going to be a controlling RNA species for gene expressions. Now we're into the world of epigenetics. So the repeats themselves, those untranslated repeats, undergo an A to I, so that's an adenosine to an iosine hyper-editing, it's called. This is all epigenetics. Results in a CTN binding to a protein called DBHS, a class of DBHS proteins, and you get a retention of it in that subnuclear structure called the perispeckle, and there it is retained. There is your RNA species bound to this protein. All right, now, Drosophila behavior, human splicing, that's DBHS, family proteins. That's what this protein is. This is one type of it here. This P54NR beta is an DBHS family, a part of that family of proteins. They're associated with transcriptional regulation, RNA processing, and transport, and DNA repair. They do a lot of things. That perispeckle does a lot of things in controlling what comes out of the nucleus via all of those mechanisms, right? Regulates transcription, regulates RNA processing, and regulates transport, even regulates DNA repair. Okay, so thus they are event ontological agents. They're happening through time. They're controlling something, all of those processes. Right? So what kind of processes? They mediate protein-protein and protein-DNA, protein-RNA interactions. And so they are emergent. They're as an emergent equipoise, okay? So they're sitting there poised, ready to respond to something. And they're subnuclear molecular infrastructures, right? And so that's my writing of this paper published in Nucleic Acid Research, which talked about those proteins. All right, so that's where I get all this literature from. So I take accompanying literature and I try to explain to you what's really going on here. You need to do this. You need to read the literature. Read the literature. So if you get now those repeats, as I told you, we do this hyperediting. Now check this out. If you get a stress response, which generates things like pro-inflammatory cytokines, or indeed antigens like LPS, which binds to the toll-like receptor 4. If you get any of that happening, right, on these cells, you're going to get a signal. We're not too sure yet what that signal is, and that's going to cause a cleavage of that otherwise sequestered MCAT2 polypeptide, the RNA for that polypeptide. It's going to cleave that. It's going to leave the perispeckle. And now you got an MCAT2 protein, again, now synthesized, but a lot of it, because a lot of it was sequestered, the RNA was sequestered. And when enough of that builds up, it binds to this membrane here, and it induces nitric oxide synthase. Now you get an increased production of nitric oxide, and that's the whole cellular stress response that's induced by this system. So you see how this could be coupled to what we just talked about with losing perispeckles. If you don't have that regulatory component there, you could potentially take all of this CTN RNA and either degrade it and have no regulation after a stress, or you could build it up without being sequestered here in the perispeckle, so that it immediately goes to make more MCAT2, right? This gene product, that cation transporter protein, opening up the cell, causing all kinds of signaling and all kinds of communication with whatever that cell happens to be, something in the central nervous system, obviously, okay, like a oligodendrocyte. And then that could increase or enhance a superinduction of nitric oxide, and that could lead then to oxidative stress and what? Neurodegeneration. See? It's good to know this stuff. So let's make a synopsis of this NEAT1 and schizophrenia as of 2019. All right, call it a day, call it a morning. All right, so let's take a look at this. NEAT1 and perispeckles may activate the innate immune response via the sequestration of RNA and DNA binding proteins. They actually do that. So some of those proteins that end up on that scaffold, that protein scaffold that's found in the perispeckle, can prevent things like gene expression, because if you bind up RNA polymerase, or you bind up ribosomal RNA, or you bind DNA, right, you're going to get an epigenetically altered transcription, right? So it says here, thereby epigenetically enhancing nascent vectorial transcription. So if you sequester enough of the proteins that bind to DNA and RNA, you could flip it either way, the valence between generating more transcripts or less transcripts of specific structure, right? Based on where these proteins are bound to, what RNAs are bound, the specificity may lie in there. So NEAT1 also regulates the oligodendrocyte differentiation, and it does so via these means. This paper we just looked at demonstrates that there is a downregulation of NEAT1 in the cerebrocortical regions of schizophrenic patients as compared to healthy with controls. Now, these are going to be post-mortem studies. The NEAT1 concentration drop associates with reduced oligodendrocytes and the sphingomyelin biostatic machinery. So they also looked at this. So see, now they're looking at more and more detail. They're looking at how things interact. They're generating an interactome. The paper's much better than the first one we covered. And they also looked at mouse models to back it up. So you've got autopsy, post-mortem analyses of schizophrenic brains, and they're backing up doing a double knockout of a NEAT in mouse to determine whether or not what it's altering looks like a sphingolipid biosynthesis. So we know now that some of the proteins that are involved in that perispeckle mediated control over transcription are indeed the genes that code for the proteins that are involved in sphingomyelin synthesis. Very interesting. The data provides evidence that NEAT1 epigenetically modulates oligodendrocyte transcription patterns, definitely. Emphasis that this is post-mortem research, okay, I want to emphasize that, on diagnosed patients, and thus poses the usual caveats to providing in concreto evidence linking NEAT1 as either necessary or sufficient to drive schizophrenia. So again, these are diagnosed patients. They presented with schizophrenia. They do have this NEAT1 suppression. They do have this alteration of oligodendrocytes. We do find this change in transcripts when we do the knockout in mice for NEAT1 to compare it to what we're getting in the post-mortem tissue, right? But again, you're comparing animal studies with human studies, and again, the human studies are associated with post-mortem analysis. But still better, still much better than the first paper. Log's better. So anyways, what I say, however, combined with the mouse model evidence, NEAT1 does seem to control the oligodendrocyte-mediated neuronal sphingomyelination. So that's important to know. Perhaps the evidence on NEAT1 dysregulation reveals a larger molecular landscape for the role of the long non-coding RNA-mediated epigenetic reading, writing, erasure in neuroimmunology. So what are some of the implications using my paradigm of dia-event ontology? Single biochemical or cellular events cannot be trusted to explain complex neuropsychiatric disease. Animal models, while indispensable for things like pharmacotherapy and pharmaceutical discovery and just modeling diseases, will not apprehend, in my view, neuropsychiatric disease, because neuropsychiatric disease is a human disorder. Systems approach, such as standard omics platforms that would be DNA, RNA, protein, lipid, right? Lipidomics, transcriptomics, proteomics, things like that. Or even cell sorting screening using cell sorting instrumentation from biological specimens fails to imagine the three-dimensional architectonics of genetic, environmental, and neuroimmunoepigenetic phenomena as event ontologies through time. There should be an event ontology. So I say a paradigm shift is necessary to approach and indeed engage neuropsychiatric disease. And I'm claiming that that's what we need to do. We need to use these tools, but we need to recombine them in ways to design experiments to be able to get at the heart of these very complex, very debilitating, morbid, sometimes mortal, causing death diseases. So let's take real briefly a look at the dialectical method. Phase one is start with a dialectical approach that transcends dogma and conventional principles while preserving truths via coherence and foundational ascendancy. Isolate and verify premises that can be used to generate sound epistemological arguments, ones generated to find knowledge, right, like through concepts. Then generate hypotheses via the deductive method, implementing careful hermeneutical analysis of both cross-sectional and indeed cohort-based published research. Use both of those kinds of research. produce theses, antitheses, and syntheses according to a justification of truth, qualia, that better explains the necessary and sufficient relational competency of rational foundational concepts. All I'm saying is that before you ever get started, you should carry out a very careful dialectical analysis using the literature and using the power of reason. Right? So this is both epistemological as well as pure reason phenomenon. That's how the brain works. So then we have to uncover the unbent ontologies. That's phase 2. You start with concepts that are obtained through temporally centered experimentation, like what we just saw in that paper. Except, of course, it was post-mortem. It would have been nice to have living subjects. We'll get that in a second. Avoid animal models as primary source of evidence, ideas, or concepts. Comprehend biochemical interactions as vectorial, dynamic flux within cells, tissues, and organs, and entire organisms, and recognize that those processes have 3 phases, at least: initiation, extension, termination. Design experiments that are non-invasive involving living subjects via new methods in analytical spectroscopy, scanning technology, and indeed molecular pattern imaging, using, for example, fluorescent antibody tags. Verify all the evidence back to published literature all the time, reliability of the technique being used, and reproduce each experiment several times through time, in time. Finally, what we need to do is improve our concepts and approaches. That's phase 3 of this paradigm shift. Develop biochemical and physiological ontologies for the human system. Avoid brute skepticism, but embrace the uncertainty of research. Discover and internalize verified evidence from empirical and rational sources to produce personal metaphysical ownership of ideas and concepts. So in order to do this, we have to work on individual patients. And we have to get a much better history of them to get a better idea of how their mind might function when they were healthy, so that we understand how the abnormalities perhaps fit the endophenotypic changes in the biochemical pathways. See? So we have to differentiate these diseases as well. And I'm going to leave that up to the psychologists. Right? We should also very much avoid in concreto dogma in favor of a working intellectual dynamic. We have to be there using a working intellectual dynamic, processive. So that means I'm just about done here. VerEvMed, Authentic Biochemistry is where I'm from. Those are my companies. I co-own VerEvMed, and Authentic Biochemistry is just mine. It's a new podcast. There's the email address for the company. There's the website for the company. Please come talk to us. We are scientists verifying published evidence in medical biosciences. So we can take anything you're interested in, like right here we're talking about schizophrenia. I can dig in the literature for you. I can work to generate something like a PowerPoint like you saw, a video like this, or we can work one-on-one, have conferences, and generate as much information as you need to try to get, for example, if you're designing experiments, or if you want to get a good literature search and you want to get rid of all the hubris and hyperbole. That's what I do best, right? Read, interpret, verify evidence, and then synthesize all those components into coherent understanding of the largesse of the literature. Again, there's the Facebook page. We get all these free videos and a lot about how to get to VerEvMed and indeed Authentic Biochemistry. There's my individual email address. You can contact me. I will usually respond within 24 to 48 hours after each of these videos. Again, that's me, Dr. Dan Guerra. I was really glad to present this to you today. And what I always do at the end of these lectures is to say my sign off, which is bye for now.